hear me? Hello, hello. Nice. Okay. So, I like to call myself a quantum engineer. This means that I build apparatuses to study and control objects that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics as opposed to the laws of classical mechanics that rule everything big around us. So you might find it a little bit of a shock that I study things as birds and butterflies. But bear with me. Before I explain where I'm going, I really need to tell you where I come from. We're going to start talking about small things coming from quantum mechanics, and we're going to end up talking about things of biological relevance, such as organismal migration, how the cells in your body respond to radiation and oxidative stress, and how we might use strategies employed by nature to build better technologies. And that's because I am a quantum engineer interested in how quantum physics informs biology at the nanoscale. So, I'm going to start my journey by saying that humankind is sort of obsessed with measuring things better because this means that we understand them better. So we can think about measuring better frequencies with this atomic clock that sits at NIST. NIST is the National Institute of Standards and Technology, and we use such clocks. This sort of machine is used to define what a second is very, very precisely. We can think about measuring better magnetic fields so that the image of your baby is better resolved. So this is the magnetic resonance image of a baby inside the mother's belly. And also, we could think about measuring better acceleration so that your gaming experience is improved. Such tiny accelerometers are found inside your phone, inside your game console. But the question that I ask is, what happens if the quantity that you want to measure is very, very small? Or worse, what happens if the object causing the quantity to be measured is very, very small? I'm going to argue that in this case, you need a tiny little sensor that's able to sense tiny little quantities. If the sensor is tiny, let's make it really, really tiny. Let's make it quantum. There's a whole body of mathematical arguments that say that if you use a quantum object to measure things as a sensor, your measurement is improved. In other words, the sensor quantumness enhances the measurement. Okay? So this forms the base of the field that is known as quantum sensing, but it's also the base of the field called quantum computing, right? And you saw in Adam's talk this picture of the quantum computer at Google. So let me give you a tiny little introduction on very, very tiny, on how quantum computing and how quantum sensing work. They're both based on the unit of a quantum bit. The quantum bit is a unit of a quantum computer or of a quantum sensor. One type of a quantum sensor, of quantum bit, is a spin. And a spin is a very tiny thing. So, thank you. A spin is a purely quantum mechanical property that has to do with how particles interact with magnetic fields. Electrons have spins, and both there and here you see a quantum mechanical representation of an electron. Okay? This electron has this property called spin, which is represented by this arrow, and the arrow can be up or down, which by this, physicists only mean that this quantum property called spin has two energy states. Okay? It's called spin because physicists represent this as one electron spinning either clockwise or anti-clockwise. Yeah, you can do it. Awesome. Okay? And it turns out that those two different spin energy states makes your quantum bit. Okay? A classical bit can be a switch, can be thought of as a switch, 0 or 1. In your computer, 0 or 1 are encoded in voltages. Bit 0 means that you have, say, 1 volt voltage, and actually it's a 0 volt voltage, and bit 1 means, say, you have a 5 volt voltage. Okay? a quantum bit is sort of the same. It also has two different energy states, but with a very important difference that a quantum bit can be both at state, in one energy state and the other, and you can try to do. And this is called a superposition, and this property is behind the field of quantum computing and quantum sensing, and it gives many of the advantages that we see experimentally. Give it to the spin here. Thank you. So, but I was telling that the sensor quantumness enhances your measurement. And I'm going to start talking about a technological quantum sensor, and that is a spin in the material, diamond, that we have in our rings, okay? And one can put a spin in diamond to work as a very sensitive quantum sensor. And that's the way that it works, right? So the spin that I'm talking about arises from a crystal defect in the diamond lattice, and this spin is actually it's called a color center in diamond. This only means that it absorbs light and it emits light, and this light is known as fluorescence. Color centers are the things that are responsible for the color of those beautiful diamonds that you see there. The way that we play with those things in the lab is it's all done at room temperature. So we take a tiny slab of diamond. Actually, the first diamond that I played with was given to us by a jeweler. So you put this slab of diamond on a moving stage, okay? And at the same time, you put it under a microscope and you shine light at it. The moment that the light hits one of those defects, one of those spins, it's going to absorb light and then fluoresce. And when it fluoresces, we can capture the light emitted by it. So that's what it looks like in increasing zooms. And at the bottom panel there, what you're looking at is a blob. It's a blob of light emission that we see. And it's super cool, because what you're looking at is actually the fluorescence of one single spin. As if it weren't, I mean, it's incredible, but it gets even more exciting than this, because just by looking, and I actually need a spin, just by looking at how strongly that blob, thank you, the spin comes back. I just need one. Okay, no, you can, yes. Just by looking at how strongly that blob is emitting light, we can actually know if the spin is in which energy state it is, down or up. Right, so just by looking at how strongly we're collecting fluorescence from that spin. Thank you, spin again. Okay. And this spin can be put to use as a very sensitive magnetometer in the following way. Those are the two energy states represented by the spin up and spin down. Sorry, I didn't call you this time. And it turns out that one of those states is insensitive to the presence of magnetic fields, whereas the other one is sensitive to the presence of a magnetic field. And just for you to have an idea, the energy difference between those two energy levels is on the microwave range, okay? So what happens is that in the presence of a magnetic field, the zero state there, one of the spin states, doesn't care, whereas the other one is going to change its energy. So here's the cool trick. Measuring a magnetic, and this energy change is actually proportional to the magnitude of the external field, so that measuring a magnetic field is actually mapped onto the problem of measuring a mismatch delta from a known difference in energy. And this is something that engineers and physicists have known how to do for decades. It's a basic problem of signal processing. So that's how we put a tiny little quantum object to use as a magnetic field sensor. And the vision is to have a tiny little tip containing one of those spins that we can bring into very close proximity to a sample, so that it can measure tiny little magnetic fields caused by one or a couple of nearby atoms. Okay? So that's remarkably cool. But then again, I'm not here to talk about spins in diamond. I'm here to talk about something way cooler. So until recently, those were the type of sensors and quantum sensors that I was working with. But actually, I soon realized that nature, many, many times, has developed sensors that outperform humankind-made sensors in awesome ways. I'm going to talk now about a nature-made magnetic sensor. But in order to talk about it, I'll have to start with biology at the nanoscale, namely the chemistry. Okay? So it has been known in chemistry for many, many decades that magnetic fields can alter the final products of a class of chemical reactions that are spin sensitive. And I'm going to call our spin, I just need one spin. So it turns out that some chemical reactions, right, at some point while they are occurring, they get spin sensitive in the following way. Basically, the reaction can take two different pathways depending if they spin, a certain spin is up or down. Okay? This means that external magnetic fields that can actually alter the probability that we find the spin up or down is going to influence the branches that are taken in certain chemical reactions. Importantly, the final products of those chemical reactions, depending on which spin branch was taken, those final products are different. Okay? So it's in this sense that a finicky quantum process, such as spin going from up to down and actually changing as a function of an applied magnetic field, can have macroscopic consequences, such as different final products in a chemical reaction. Okay? This has been, and thank you. This has been demonstrated at room temperature, in the solid phase, in the gas phase, in the liquid phase, and for magnetic fields as tiny as the magnetic field of the Earth. In the beginning of the 2000s, biophysicists were musing on how organisms such as birds, turtles, and butterflies could actually migrate. And it's known that they migrate using, at least as a partial cue, the magnetic field of the Earth, which is weaker than the magnetic field of your cell phone when you hold it like this. So how might they do this? So there was a hypothesis that they posited, which was that were the same sort of spin mechanism that I just described, were it to be possible that such mechanisms were happening inside cells and organisms, maybe organisms could sense magnetic fields to the extent that they could sense different physiological concentrations of products coming from such spin-sensitive chemical reactions, right? So, and people started proposing, right, and this is a simulation, that maybe, for instance, there was something in the retina of migrating birds that that, depending on magnetic fields, would say modulate the total light seen by the birds, so that the bird, just by changing its head, would see superimposed to an image a certain modulation of, say, of light intensity, and that could tell them where to go or where not to go. Okay? It turns out that biophysicists then started looking for things. They started looking in birds, where which sort of protein in the eyes of birds could sustain chemical reactions that were spin-dependent. And they found one, at least. And this protein is called Cryptochrome. It is a photoreceptor, that is, it absorbs light. And it's present in the retina of migrating birds, in our retina, in the antennas of migrating butterflies, okay? But it's also super important because it's present in all our cells, because it has circadian rhythm regulation functions. And whereas this picture here of the bird looking at different ways and seeing different intensity modulations of light, while this still remains a hypothesis, right? magnetosensing in a way that is mediated by this protein called cryptochrome might be very widespread across the tree of life. I plot there many different organisms that are known to express cryptochrome. You see there's bacteria, there's plants, there's insects, right? And there's mammals, and boxed there, you see organisms to which there has been some sort of experimental evidence that magnetic field sensing is correlated with the presence of cryptochrome. And if you see that, humans are right there. So evidence for this type of cryptochrome-based magnetosensing is very widespread, except at disconnected length scales. On the very short length scales, at the chemistry level, seen in this column to the left here, chemists have done experiments with vials of these proteins in solution, right? And what they see, it's very, very nice, because what they see, first of all, is that the fluorescence intensity of this protein is also magnetic field dependence. And here, I would like to bring to your attention the one-to-one correspondence with what I just described about the spinning diamond. Just by looking at how strongly that spin was emitting light, we could know if the spin was up or down, right? Here, it's sort of the same. Researchers in that top left plot there, what they did was they recorded the light emitted by those molecules as a function of the time during which you excited this molecule with light. And the first thing that you see is that fluorescence signal is decreasing. That only means that the protein is, the sample is actually dying, is being degraded, but that's fine. But as researchers post a magnetic field on and off at the same time that they were acquiring this info, they saw like a modulation of the intensity that went up and down according to the magnetic field. So what we're doing, they were doing there was some sort of quantum state readout of the spin of that protein. All of that at room temperature, right? On the other part of the length scales, we have a lot of experiments done, say, in birds and other big organisms, right? For instance, birds during migration season, they want, if you put them in cages, they want to migrate following like a particular direction. And it turns out that if you put coils around their cages, you can actually mess up with the direction where they want to get out from, right? Flies. Flies do not migrate, but they can be put to, they can be trained to sense the presence of food via the presence of a magnetic field. I have no idea how they do this, don't ask me. However, when researchers knock out this cryptochrome gene from the flies, that is, they remove the ability of the flies to make this protein, the flies can no longer be trained to find food via the magnetic field. In a further experiment, the researchers put back inside the flies that didn't have fly cryptochrome, they put back human cryptochrome. And then the flies were back to finding the magnetic field. It's pretty cool, right? Except for the fact there is no unambiguous evidence that whatever is going on chemically is actually driving physiology. Because, I mean, it's too big, the step from a vial of molecules to a whole bird. And that's exactly where our group at UCLA comes in. We want to bridge those length scales. We're building powerful microscopes where we can see, have a look at one fly cell or one cell from a bird's retina and try to perform this sort of chemical experiment, but inside living things. Okay? So, and this whole story is actually not, it's not only about navigation and how organisms can follow the magnetic fields. The same sort of spin-dependent chemical reaction mechanism might be at play in a way that is more widespread than thought. For instance, many articles have come up that actually correlate magnetic fields with how cells grow, how cells regulate the presence of harmful things called reactive oxygen species, right? And just I want to talk about one quick experiment. So bio people have this black box machine called a metabolic analyzer, and they put like plates of cells inside, and then they read out some sort of metabolic parameter that says how well the cells are doing. Physicists shielded against external fields such a machine, and with a plate of cells inside, they applied tiny magnetic fields inside on the order of the Earth's. And only by changing the magnetic field direction, they could see differences in reading related to respiration rates and related to the acidification medium of the cells, in a way that is actually consistent with the same sort of spin mechanism that I just told you about. So that's pretty cool, right? So the question that my group and other people are trying to answer is, can quantum physics be established or refuted to account for a relevant biological phenomena, and importantly, be manipulated, be controlled for technological and therapeutic advantage? For instance, I'm going to give you 3 examples. For instance, if we understand how organisms interact with magnetic fields, with radiation, and if we're going in a place with radiation, can we take a drug that actually messes up with the spin mechanism so that the pathway is sort of blocked so that we're immune to radiation? Second thing, can we start applying magnetic fields in controlled ways so that we tweak metabolism, so that we tweak the way that the cells work, so that we try to correct for, say, metabolic disorder, right? And 3, what I have just shown you is that there's evidence that organisms are for a short time behaving as living quantum sensors, or if you want, or like a tiny unit of a quantum computer. So can we learn with those strategies and maybe develop ways of doing quantum computers, like quantum computing at room temperature, in a way that we don't need this big refrigerator that Google uses, right? So I'm proposing that we actually augment this field of quantum sensing with this living quantum sensors, right? Because all of those are phenomena that are quantum, that happen at room temperature and in relatively messy environments. This field is called quantum biology, and I'm not endorsing this scientific journal called Nature, but I'm endorsing Discover, and I really think that by joining forces, we actually can solve a lot of more interesting problems than if we just are restricted to our own disciplines. And next time we play, I want to be Doc Quantum. There's Doc Quantum there. Okay, so my name is Clarice Aiello. I lead the Quantum Biology Tech, or QuBiT lab at UCLA, and may the quantum be with you, MindshareLA.